Hello, I'm Dr. Qun from the Oncology Department of Shanghai East Hospital, Tongji University, China. I'm glad to be invited to this year's World Breast Cancer Conference. Due to a time conflict, I'm unable to attend the conference on site. I deeply regret that. I would like to congratulate the conference be successful convent and hope everyone can benefit from the meeting. Due to my avatar blocking some of the content of the PowerPoint presentation, I will turn off the camera during the formal lecture. Please understand it. Thanks a lot. Okay, so my topic today is the delta subunit of GABAA receptor is necessary for the GPT2-promoted breast cancer metastasis. We know that tumor cells have a preference for glucose and glutamine. Compared with glucose, glutamine not only provides nitrogen but also carbon, playing a crucial role in the occurrence and development of tumor metastasis. Our previous research found that GPT-2 can reduce the formation of alpha-ketoglutarate in breast cancer cells. GPT-2 convert between glutamine and alpha-ketoglutarate by reversal catalytic reactions. The decrease alpha-ketoglutarate after the increase of GPT-2, leading to accelerated growth of breast cancer. Alpha-ketoglutarate is abbreviated as alpha-Kg. The reduction of alpha-Kg adjusts the activity of dioxygenase, including PHD2. Therefore, makes HIF1 alpha hydroxylation reduced and stabilized the protein level of HIF1 alpha. Therefore, promoting the growth of breast cancer. From the figure along the left, we can see that colon formation increased significantly after overexpression of GPT2, while after downregulation of GPT2, there is a significant decrease in colon formation. We can see that animal experiments have also obtained similar results. GPT2 expression increase in metastatic breast cancer. We can see that the proportion of breast cancer metastasis increase in cells with high GPT2 expression. GPT2 expression increased in metastatic breast cancer cells. This result suggests that GPT2 may promote the metastasis of breast cancer. Does GPT2 promote the metastasis of breast cancer? Through the transfer experiment in vitro and the tail vein injection experiment in vivo, we found that the increase of GPT2 did increase the migration and metastasis of breast cancer. How does GPT2 promote the metastasis of breast cancer? In the previous experiment, we found that overexpression of GPT2 can reduce the content of alpha-Kg. So we all know that glutamate can not only be converted into alpha-Kg, can also generate GABA, which is not only a neurotransmitter, but also an important metabolite of glutamate. GABA is an inhibitory neurotransmitter in the brain, and it can also play an important role in peripheral tissue cells through GABA receptors. We found that or expression of GPT-2 increased GABA content through metabolic mass spectrometry experiments, indicating that GPT-2 increased GABA content by increasing glutamate concentration. Thus, GABA-mediated GPT2-promoted migration of breast cancer cells. 3-MPA is an inhibitor of GAT, which is an enzyme that generates GABA. After adding 3-MPA, GABA cannot be generated and cannot be migrated. At the same time, overexpression of GAT1 promotes migration of breast cancer. The overexpression of GAT1 also partially restored the migration of breast cancer with GPT2 depletion reduction. These observations suggest that GABA as a single molecule mediates GPT2 to promote the migration of breast cancer cells. The binding of GABA to a GABA receptor can lead to changes in cell membrane ion permeability. GABA receptors mainly include ion channel type GABAA receptors and metabolic type GABAB receptors. The different physiological functions of GABA depend on the binding of different receptors. Which receptor does breast cancer depend on? GABA receptors include pentamer, that are 2 alpha, 2 beta, and 1 gamma. Each alpha, beta, gamma is not a single protein. They all have family members, among which gamma includes 5 family subunit. GPT2 induced cell migration is dependent on GABAA receptor type receptor. Picrotoxin is a GABAA type receptor inhibitor, and the CGP52432 is a GABAB type receptor inhibitor. The migration ability of breast cancer cells decreased after using type A receptor inhibitor. After the use of type A receptor inhibitor, even if GPT2 is expressed, the migration ability of breast cancer cells is still reduced. The beta subunit of GABA includes 3 family members, BET1, BET2, BET3. We can see that knocking down BET1, BET2, and BET3 are family members, even when GABA and GPT2 are expressed. The migration of breast cancer cells is also significantly reduced. Transfer assay was used to detect the effect of GABA on cell migration in breast cancer cells with GABRA1, GABRD, GABRP, GABRQ, or GABRG3 knocked out. It was finally detected that GABRD knockout had the greatest impact on migration and eliminated GPT2-induced migration of breast cancer cells. In lymph node positive patients, the higher the impression of GABRD, the poorer prognosis of the patients. So we can see that gamma subunit delta members are essential for GABAA receptor mediated migration of breast cancer cells. Through gene counts, we found that the calcium ion binding protein pathway has the strongest activation ability. The GABA receptor activates the calcium ion channels mediated visit, causing calcium ions to flow in. Our experiment found that overexpression of GPT2 resulting in an increase in intracellular calcium ions. After knocking down GPT2, intracellular calcium ions decrease. After adding GABAA receptor inhibitors, intracellular calcium ions were inhibited. After adding GABAB receptor inhibitors, intracellular calcium ions were not affected. After adding calcium ion chelating agent, intracellular calcium ion and the migration ability of breast cancer cells decreased. Even if GPT2 is overexpressed, the migration ability of breast cancer cells is still decreased after adding calcium chelating agent. Therefore, calcium ion mediated the migration of GPT2 or GABA enhanced breast cancer cells. After adding calcium ion chelating agent, intracellular calcium ion reduced and the migration ability of breast cancer cells decreased. Even if GPT2 is overexpressed, the migration ability of the breast cancer cells is still decreased after adding calcium chelating agent. Therefore, calcium ion mediated the migration of GPT2 or GABA promoted breast cancer cells. CREB activation is the key to GPT2 or GABA-induced migration of breast cancer cells. GO analysis revealed an increase in cell adhesion pathway. The phosphorylation enrichment experiment found 151 phosphorylated transcription factors, which intersected with 184 transfer-related protein found in the database. And 5 phosphorylated transfer-related proteins were found. Among the 5 proteins, CREB protein was found to be the key to induce migration of breast cancer through a phosphorylation enrichment test and total protein phosphorylation test. CREB protein expression, even if GPT2 is knocked out, does not affect the migration of breast cancer cells. But after using CREB inhibitor, our expression of GPT2 results in a decrease in cell migration ability. There are literature reports that protein that directly bind to calcium ions include PKC, CAMK2, and pKa. There are also literature reports that PKC regulates the activity of CREB. We confirm that the potential kinase of the CREB is PKC through immunoblotting. A volcano map showed that the most up-regulated and down-regulated genes of GPT2 and analyzed the gene expression profile between the control group and GPT2 overexpression breast cancer cells. The first 5 GPT2 up-regulated genes were verified by qPCR analysis, and the CREB inhibitor 66615 can block the expression of GPT2 up-regulated genes. We can see that CREB promotes breast cancer metastasis by up-regulating MMP9 and the other genes. We found that metastasis still occurred after GPT2 knockdown and GABA overexpression through 2 photon in vivo imaging experiments. After the application of GABA inhibitors and CREB inhibitors 66615, the metastatic ability of GPT-2 overexpression mice decreased. In order to investigate the effect of GPT-2 on breast cancer metastasis, we first introduced a GPT-2 gene knockout C57BL6 mice, and then mated with MMTV PYMT mice to generate MMTV PYMT. And GPT-2 plus minus and MMT PYMT and GPT-2 minus mice. As shown in figure B, the knockout of GPT-2 to some extent extended the overall survival of tumor burden mice compared to the wild-type breast cancer mice model. In the mammary gland conditional GPT-2 minus mouse model, GPT-2 knockout significantly decreased long metastatic nodules and prolonged the overall survival of tumor burden mice. The immunohistochemistry staining analysis also showed that GPT-2 knockout markedly reduced GABA synthesis, PKC and CREB activation, and MMP9 expression in mouse breast tumor. According to the previous experimental results, we can conclude that GPT-2 increased the production of GABA in breast cancer cells, and GABA promotes the metastasis of breast cancer, mainly through PKC to regulate CREB phosphorylation, further promoting the tumorogenesis and metastasis caused by the activation of MMP9 protein. It should be noted that delta is the key subunit of GABAA receptor mediated breast cancer migration, and the CREB activation is the key to GPT-2 or GABA-induced breast cancer metastasis. This is the personnel who completed our article. Junmi and Qunli are the corresponding authors, and Nali, Xiangxu, and Dan Liu are the first authors, which balanced contributions and no conflicts of interest. That's all for my lecture. Thank you for listening and welcome to ask the questions. We will discuss the question section together. The last corresponding author, Professor Junmi, will also communicate with you online. Thank you.